maatschappij te gebruiken.
Okay, thanks for having me. Can the technicians also put on the slide on the lower presentation? Okay, so it's a bit odd presentation in the sense that it's a combination of more the medical stuff, but also I want to dive a little bit into the technicalities. So sometimes I have to hold back with the technicalities at the hospital, but now I can go all out. So this is briefly the outline of my talk. So I'll first explain a little bit about cancer. Yeah, the presentation is coming up. Thanks. About cancer and about mutations, and then I'll dive a little bit more into the methods that are used to analyze this stuff. And I will also like to emphasize the uncertainty aspect. So when we're in the hospital, we care about uncertainty in the sense that we have to make decisions, and it's important to know when the model is being not certain about its prediction, or when it is certain, so that we tackle using Bayesian analysis. But I'll get to that when we get there. So I want to just first take a step back. What is cancer? So frankly speaking, I still don't really know what is cancer. There's just such a complicated topic. So the definition I put here is actually just I took it from Wikipedia. And so there they write it's just uncontrolled growth, and it's particularly uncontrolled growth in the sense that it tries to colonize other parts of your body, and it tries to invade neighboring tissue. So that is basically the description of what you see. But at a more biological level, there are a few what they call hallmarks that drive this disease. And so there are a couple of them, so I'll just try to be brief, but I think it's nice just to get some intuition what is cancer and how are they things related. So one of the things is that they somehow figure out how to prevent the cells from dying. Also, they need a lot of uptake of nutrients and oxygen and so on. And so they managed to find a way to build blood vessels. So this is called angiogenesis. Each cell has its own countdown clock. When every time the cell is renewed, then the countdown decreases. And so they also figured out a way to reset the clock. And then invasion and spreading, so this is already in the definition you say from Wikipedia. And then growth signals are also important. So these two definitions, self-sufficiency and insensitivity to anti-growth signals, look very related, but at the biological level, they are actually different parts. So they try to, when other cells say, stop growing, they figure out a way to not do this. And, they also have made a way to get signals from themselves to keep growing. And so it's important to realize that just one of these characteristics is not enough. You really need to get all of them simultaneously. And usually this is acquired using small letter changes in the DNA. But also other parts are important, but I will not focus on this talk. So this was really just a brief introduction for cancer, so that you have a more concrete idea of what we're talking about. So how do they use AI in the clinic? So getting stuff in the clinic is quite complicated, but there's, from the science part, there's lots of stuff going on. For example, here in the UMCG, there's one of the few places in the Netherlands where they have a proton center, and they radiate the tumor with small protons. And so they need to figure out what to irradiate, and so models do that. And so AI is now also doing the calculations, what to irradiate to to choose the right spots. I'm personally quite excited about single-cell sequencing. So you can actually just take out cell by cell and then read off the DNA and the stuff that's in there. And they use small microfluidic devices to sort all these cells, and then they can measure them one by one. It's really amazing. I think one aspect which is already quite advanced is detection of small cancerous lesions in what is called CT scans. So you make a chest scan, and then you have the AI look at suspicious so-called nodules. And these AI tools are really great. They outperform actually trained doctors. The pathologists, they put small slices of the tumor on the microscope and they look if there's any cancer in that. And of course, AI can also help detect this stuff. Usually AI is great when you have images, so this is an easy task to do. And I'm also just flabbergasted by the protein folding developments, where you just have a few amino acids chained together, and then it just outputs the protein structure without even understanding the actual physics that go behind it. It's just mind-boggling. And now there are also new developments that stack on top of that to see what, if you change some of the amino acids, what happens to these proteins, and does it help in predict disease? And it turns out the answer is yes. There are also developments in the liquid biopsies, which is just a way where you take some blood and then just analyze it. Yeah, there's lots of DNA floating around, and yeah, you can imagine that you can also find patterns in there to help predict treatment outcome. And that's something I've also worked on myself. And the topic of this talk actually will be mutations in the cancer. So it turns out that all these characteristics I discussed previously, most of them they acquire by changing bits of the DNA. And this will look into more detail. So just a very brief introduction about DNA. So I think we're all technicians, and myself, I also had very little biology background a couple of years ago. So I want to take you a step back just to explain what this is all. So your cells, they all have a bunch of chromosomes, and they are actually just very large strands coiled up around small proteins. And when you unpack this, then it turns out that this is just a sequence of letters, and some parts are coding, and these are called exons, and some parts are non-coding. And this entire sequence are made up of A, C, T's, and G's. And there are different parts where you can create mutations. So you can just duplicate entire chromosomes. You can remove small regions, but you can also just have small single letter changes. And this is actually the part that we will be focusing on, because there have been lots of developments in measuring these letters. And this is done using a technology that is called next generation sequencing. And in essence, it's just a fancy microscope. The trick to read off these DNA is by first having these letters, making them fluorescent, so they have a color, so that they hybridize like they show in the picture. And then, you just make a bunch of the same copies, and then when you take a picture, then you see actually a blue dot. Well, then you attach the next letter, make another picture, and then just stack them on top of each other, and then actually you read off chunks of DNA. It's just very clever and amazing that we can just really read off this DNA in this way. So I'll be looking into mutations, and specifically single letter changes. So I'm sure you know that when you go out in the summer during the sun, you have to put in some sunscreen to protect yourself from the sun. And we all know that smoking is bad, of course. And the reason is because they cause these small changes in the DNA, which makes it more likely that you will end up with cancer. And it turns out that it doesn't happen just uniformly across the genome. They have a kind of preference. They like to sit in specific sites. And that actually depends on the type of damaging agent that makes the letter changes. So for example, sunlight makes very different changes depending on their neighboring left and right context compared to, for example, cigarette smoke. And this is actually quite clever because it means that we can infer how these mutations were created by looking at the local motifs. So here, for example, I have shown from cigarette smoke that you can really see that they like to change the C to A, and they have some preference from the neighboring letters. So just by looking at what kind of mutations you have, you can infer how it was created. So they call this genomic scars because they leave this kind of scar which tells how they were created. So how do we analyze this? Well, we use non-negative matrix factorization. And just the too long didn't read summary is this equation. Yeah, so it seems very simple, but I think it's quite powerful technique. So you factorize it in two matrix, and as the name suggests, everything is non-negative. And that means that it will learn a sum of parts because the elements cannot cancel, so they also have to add on top of each other. All the elements have to be non-negative. And so an example is here an image where they apply this non-negative matrix factorization. And then on the left you can see the part that the NMF has learned. So these are the W, the weights. And then you can see that it learns parts of the eyes and parts of the chin and some of the contours, and they all add up additively to reconstruct the image. So that is the basic part of non-negative matrix factorization that makes it so attractive. But there are also other ways of looking at this model, different perspective essentially, and I want to go over them very briefly. And another way is that it is an unsupervised method. So it learns the distribution of the data. So X here is the input data, so for example the images. And I'm sure you're familiar with clustering methods like k-means. So they assign samples to a specific label. And this is called hard clustering in the sense that each sample gets only one assignment. And non-negative matrix factorization, you can actually imagine it as a soft clustering. You can assign each sample to some part the red cluster, some part the blue cluster, and this is very powerful in the region you see in the middle, where it looks a bit like a red cluster, but also like a blue cluster above. And NMF tackles this in a way by saying, well, fine, you're a little bit red, you're a little bit blue. And this is called soft clustering. Another perspective on non-negative matrix factorization is actually collaborative filtering. So I'm sure you know Netflix, where when you watch a video, you can give it a thumbs up or thumbs down. So you rate the stuff you've seen, and you can use that. The information of others you can use to infer what the user would like to watch. And so, for example, here I've shown a matrix where the users have seen 5 movies, M1 to M5, and they've all given them different ratings. And by factorizing it, what you end up with is the weights that's on top, and so this learns yeah, clusters of movies that give high ratings. So then you end up with a comedy and an action signature. And then you factorize it in the other matrix is the amount of contribution to these specific themes. So that is just the same thing earlier, this matrix factorization. But this they can Netflix uses, for example, to they use a slightly more advanced technique, but this is the basic principle. And so this is what we also do for analyzing these mutations. So again, we have users. So these are here these people in red, green, blue, and we count how many of these mutations occur in the neighboring context. And then we factorize it in these different components. And it turns out that these components, they represent something that exists in real life. And so, for example, these smoking signature on the right, they have this preference for specific letter changes. So you can factorize it in this way, and people have done experiments to figure out, what do these components actually mean. And then it turns out that, for example, then one is related to smoking, some is related to age, because your DNA is damaging constantly throughout life, and so on. And now I give you my fourth perspective on non-negative matrix factorization, which is neural networks. So this matrix factorization can also represent as a simple shallow neural network. And on the right-hand side, you can see that here the weights are the matrix on the right. So that's why I also put in the letter W, because they represent the weights. And then the attributions to the number of mutations attributed to these specific components are then the hidden units. And this gives rise to this model on the right, where then the observations are Gaussian draws from the factorization. And once you realize that this is actually just a shallow neural network, then it's also it's immediately clear that you can just generalize it by building multiple layers. And so this is actually what we've been doing lately, and we hope to publish it soon. And we make it a Bayesian, and I will go into that shortly, by assigning a probability distribution to each of these elements in the network. So the weights are not just non-negative, but they come from a specific probability distribution, and likewise for the latent states. Right, so this summarizes again what I said, that the different matrices have a grounding in this network. Okay, so Bayesian analysis, basically it boils down to this equation we see here. We can just rearrange the order of the joint distribution. And what is the point of this? Well, I'm sure many of you have trained your a simple neural network. And the way you do that often is just you take your probability distribution with your data, and then you find your parameters which maximize your likelihood. And so what you get is just a point estimate of your parameters. And so, for example, your W's. And, the point is that if your data changes slightly, then you may end up with a completely different W. And so, you don't characterize the uncertainty that you have in the components of your network. And so the Bayesian analysis is saying, we don't just solve this likelihood, we try to infer the distribution over the variables that we don't see. And we do this by just applying the Bayes rule. So this is written down there. So we ask to calculate the probability distribution over the weights and the hidden units, given that we have seen the data. And so now your estimates are now fuzzy. And so I also thought that it would be nice just to show, how does it work in practice, so that it's not just abstract, blah, 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 but that you can actually get some idea how this works. So again, we try to calculate at least take samples from the posterior distribution, which is on the left, the PW and H given X, so the weights and hidden units given the data. And in general, it's quite a difficult task to solve. And because the element on the bottom, P of X, is difficult to compute. And so what we do is we do Markov chain Monte Carlo by taking steps in a space. And concretely, we use a Gibbs sampling, which means that you start with some weights and hidden units, and then you use it to make a new estimate of your weights, also a new estimate of your hidden units given the other variables, so that you have a new joint sample. And this is just one step, and then you do it many times to obtain samples, which then, if you continue the chain long enough, you will end up with the distribution of your posterior. So I have not so many time left, so I'll try to wrap up briefly. So we also made a Python package if you're interested, so for this deeper layer. So this is a generalization of non-negative matrix factorization. Well, I won't bore you with the actual Python code, but just let me know if you're interested. And so this summarizes my talk. So there are many places where AI is transforming care. I think the only place it's not really transforming healthcare is the actual nursing part, but every other aspect will be transformed. And so we briefly talked about these mutations in cancer, and we used non-negative matrix factorization to analyze it. And I gave you these different perspectives of looking at it, which I think is nice to see how it comes about. And uncertainty is important, especially in the hospital, if you want to make decisions. And we made a package to help quantify this. And this concludes my talk, and I would like to thank you for your attention.
Thank you very much for that interesting talk. Now it's time for questions. Anyone? Okay.
Is this on? Yes. First of all, thank you for your talk. I was wondering, do you have an opinion on Bayesian neural networks versus post hoc methods like Bayesian dropouts? Because they're quite slow to optimize usually.
Yeah. So that's actually a trade-off we make explicitly, in the sense that what do we find more important? Well, the data that we use, it costs a couple of €1,000 per sample. So then, well, okay, it takes another few days to calculate. Fine, the computation cost is negligible compared to the compute cost, and yeah. So that is a specific choice we make. We sacrifice speed for uncertainty.
Right. Thank you.
Any more questions? Going once, going twice. All right, thank you very much. And here's your token of appreciation. And thank you. Thank you very much for this talk.